go live in momentarily.
We're going live momentarily. It's going to be great. We're going to live facilitate this, and we'll kick it off there. Hey everybody, welcome back out there to another beautiful afternoon with Franklin Outside. This is Dr. Rachel Dawn coming at you today with a very special episode with two of my favorite TFI colleagues. I have a lot of favorite TFI colleagues, and these are just two of them. And we're going to try something a little bit different today as we're still playing around with our microscopes. Hopefully you've been following along, along with us at home together, but a part, of course, to our nature scavenger hunt, which we've been incorporating some microscope images into lately. So today we thought we'd do a little bit of competition. Let's zoom in on that microscopic world and see if we can guess what we're looking at. I have two of my fellow TFI members here today. The first I see is Dr. Derrick Pitts. Derrick, how about you say hello, tell us what you do at TFI, and let us know why you think you're going to bring the competition today.
Institute. So I'm really glad to be here. I'm looking forward to this. And the reason why I'm bringing the heat today is because as an astronomer, we control the universe. We know everything about the universe. So naturally, I'm going to win.
Okay. All right. Some big words from Dr. Pitts there. So Dr. Pitts cut out a little bit, but he, of course, is our chief astronomer. And it sounds like he thinks he rules the universe. All right, all right. And Dr. Pitts's competitor today will be Leszczynski, is one of my favorite floor educators. Al, tell us what you do at TFI and why you think you're going to bring the heat today.
Oh yes, hello everyone. My name's Al. I'm really excited to be here today. I'm a science interpreter at the Franklin Institute. While we were still, together in the museum, you may have seen me doing live science shows or interacting on the floor. These days, doing a lot of online content, Spark of Science. I'm really excited to be joining the live crew today for this. And so I have a very special reason why I know that I'm going to win today. This is in my blood, and I have proof. I actually showed Rachel this before the competition. I had a microscope growing up as a kid, and my dad turned it into a lamp, and this sits in my living room that I do work next to every day. So, it's in here.
All right, Al. Okay, we got a real history here, family history here. Well, okay, so Al.
Not too deep, right?
That's true. It's true, Derrick. So. We've got the microscopes on one end here, but of course, Derrick has some experience with those telescopes too. So we both have a little bit of experience peering through magnifying lenses to check out the world, both here and away. So what I'm going to do now is I'm going to mute my computer screen. I'm going to switch over to my cell phone. And while I'm adjusting that, I'll leave it to Derrick to tell us what it is that he's holding up.
Bringing out the big guns this afternoon.
Okie dokie, and we should be screen sharing
you
there,
talk about microscopy.
Derrick, you froze a little bit, but hopefully you and Al can see my screen right there. Can you see that all right? I see some thumbs up. Great. So that, of course, is my beautiful dog. She is not who we will be looking at today. We're actually going to go into my camera here and into my video setting. That's my desk there. And what I'm going to do now is place my camera on top of the microscope, and we'll get our first guesses going. So this first one's probably going to go a little bit clunky while we set up. So hang with us here while we zoom in on our microscope image.
Oh, I'm so excited. Oh.
All right, here's our first. I'm going to start counting down a minute on the clock, and I can adjust as we go to make sure that you're getting the perfect image. I know that the lighting can be a little bit tricky here. So tell me what you think that we're looking at, and let me know if you want me to adjust the camera any which way.
I think I have a guess here.
Okay.
Al already has a guess. Coming in with the heat, Al. You weren't kidding.
Okay.
All right, we have 30 seconds. Final answers if you want to write those down, and I'll move us around just a wee bit so we can try and nab some new images here.
Cool.
And I can zoom in even a little bit further. As you can tell, I've been having hours of fun all alone at my desk.
What kind of magnification are we at?
Say again?
What kind of magnification?
It's an excellent question, Al. I'm going to hit pause on my screen sharing, so write down your final answers, and we'll talk about that magnification. And we should be back here. So we do have a bit of a time lag, so we want to adjust for that. So how about we provide our answers now, and then we'll talk a little bit about that magnification. So it looks like, okay, we have two answers. So Al guesses that we're looking at fungus. It looks a little bit like fungus. I think we might need to work on your handwriting there. A fungus among us. And Derrick guesses sedimentary rock crystals. Okay, love that guess, Derrick. We both have that geology background. So Al, tell us a little bit about why you think we're looking at fungus.
Yeah, so on that image, the first thing that struck me was that there kind of seemed like to be a little fuzzy. It was a little fuzzy around the edges. Like there was some thicker parts and then some fuzziness. And it reminded me of mycelium, which is the fungus that goes onto plants' roots and like spreads throughout the soil. And it's kind of real, it kind of looks meshy like that. And so that was my first instinct with that one.
Cool. Love that guess. So a fungus among us. If you think Al is right, go ahead and click that like button. Al thinks that we're looking at fungus there. You'll also see that photo pop up in the comments section. And Derrick, you thought we were looking at rock crystals. Why were we looking at rock crystals? What do you think?
Thinking about some old oatmeal I left in a pot. Looked like that old oatmeal in a pot looked like after it was there for a day or two. And so the way those pieces in the image that you had looked reminded me of that. And I thought, well, it can't be that. Okay, so I'll go to something that's a little closer. And I then thought about rock crystals. And that's how I got to sediment crystals.
Okay, Derrick, I love it. So Derrick thinks that we're looking at sedimentary rock crystals. If you think that Derrick is right, go ahead and press that heart button. So if you think Al is right with fungus, hit that like button. And if you think Derrick is right with rock crystals, go ahead and hit that heart button. Before I reveal the final answer, looks like we need to pull that image up one more time. So I'm going to go ahead and screen share one more time. And Al, you had a question, I believe, about the magnification that we were at. Is that right?
Yes. And
I would love to tell you it. If we can get our camera up again.
And I can't wait to hear about it. It's funny because when you're looking at microscope images, like you could think it's one thing at one magnification, but then you find out it's actually zoomed in 100 times. Yes. And it like totally changes what it could possibly be.
Yes, I love that you asked that question. So what we're actually looking at is 2 times magnification on my phone times the magnification of the laser pointer. Now, I don't actually know the magnification of the laser pointer, but we talked about this a few episodes ago. For those of us that have been following along, to get the final magnification that we're looking at, we'd want to multiply our lens magnification power. And Derrick, of course, knows a whole heck of a lot about lenses. Derrick, can you tell us a little bit more about magnification?
Yeah, so I'm going to take a guess here. You know, I'll use that scientific term a lot more than normal.
Okay. I think you might have had a guess.
I said if I had to put a number on it, I'd say that the number is probably
you have
80
me on
times magnification.
Edge of my seat there. You said 80 times magnification, Derrick?
I'm going to go with 80 times magnification.
80 times magnification. Well, we may actually be close to that, and I can tell you why in just a moment. I'm actually going to move my sample to the side to help explain further what it is that we're looking at. So remember, Al, you thought that we were looking at fungus. Derrick, you thought we were looking at sedimentary rock crystals, and they kind of do look a little bit like both, right? I'm switching here to a different view because now we've got some nice coloration in there, and I think that helps explain what we're about to reveal here. So what we're actually looking at here. You guys both picked up on it. You thought, Al, it looked a little bit spongy almost. Derrick thought it looked like individual crystals. Those are actually little dried up cells. We're looking at a cork sample. And the reason I wanted to pull in this particular image here is because you can actually see some wine traces that's infilling some of those empty gaps.
So cool.
It's pretty cool looking. So I wanted to start with cork because it actually has a really, really neat history related to early microscopy. So the physicist Robert Hooke was one of the earliest scientists to take a peek under the microscope and describe what he was looking at. He, as I say, was a physicist, was really interested in elastic properties. And so he chose the cork sample to take a peek at because he wanted to understand what it was about the material that made it so compressible and why it floated. What he found under the microscope, of course, that there were all of these empty compartments that were filled with air, and he also called those compartments individual cells. That's where we get the word cell from to this day. So this is sort of a science success story. Over 300 years ago, we're still using the word cell. So pretty cool there. Love that. I love using new microscopy methods. And Derrick, as you mentioned, we're probably somewhere around 100 times magnification here. I think that's probably a pretty darn good guess. Okay.
Yeah, the second you started to zoom out, I was like, oh yeah, that's not fungus.
Not fungus.
like exactly to my point about changing the perspective. It's so funny. And I also like this story about cork. Like I remember learning about that as a kid and it being one of those first things that was like, oh, cool. People just like put stuff under a microscope to find out what it is.
Yeah. And one of the cool things about cork, and Derrick, I know you're a wine aficionado, may know this, cork actually just comes from the bark of a tree. It's a type of oak tree called colloquially the cork oak. We switched back to my dog there. Sweet little image there.
I figured we tried to smell it. I figured.
Yes, of course, our sense of smell could be another useful tool.
If I tried to smell it, I'd probably recognize it.
To try and understand the year of the wine. I'm not quite sure which wine that we had left on that cork there, but yeah, cork actually comes from the bark of an oak tree, and it's called a pyrophyte, this type of oak, and that's why they have such thick outer bark rings. It's to help prevent forest fire threats. Isn't that wild?
So cool.
Pretty cool stuff. Okay, so we're zip and zip here, folks. So I think what we'll try and do now is switch to our next sample and see what we can do with it. I'm going to stay on camera for now, and we'll keep my dog Stout up there while I load up my second sample.
Love that dog. Yes.
She is a sweet, sweet animal. So do we have any other dogs among the group here?
I do not have any dogs, but I do have a cat named Choo Choo. Who has been a great friend over this time. I mean, the first couple weeks, she did seem kind of annoyed that I was around all the time.
Yes.
She was like, wait, I'm supposed to have the middle of the day to myself. But we're friends again. So that's cool.
Glad to hear it. Derrick, any pets over there for you?
That's cool.
Derrick, do you have any pets in your room?
Not today, but if we come back next week. I have a pet showing up on Sunday.
Well, an impending pet.
That's exciting.
I feel like there's a backstory here you're just itching to share.
Well, we decided that during this time, it's a good time for us to try to maybe bring a puppy into the family.
Congratulations!
I'm loving it.
And we'll do our training during this time off. It should be very
Derrick, I agree.
excited to find out.
I think it should be exciting, and best of luck to you and Linda. And we have our second sample up here so we can move along. And what I'm hoping to do is zoom in just on the edge here to pick up on that sort of razor blade edge. I'm going to start my minute countdown here, but I am going to continue focusing so we can try and get a nice sharp image there. Let me switch over to my photo screen, and we have a minute on the clock panelists, what do you think we are looking at? Oh, there's a better image there.
Writing down my answer.
Uh,
I feel like I have a guess that seems obvious, but it's going to be wrong because it feels so obvious.
No, nonsense. There are no wrong answers here. This is a group learning opportunity. We're having some fun. And viewers out there, tell us what you think you're looking at. Remember, we're going to reveal Al and Derrick's answers in just a few seconds here. If you like Al's answer, go ahead and smash that like button. If you like Derrick's answer, go ahead and hit the heart symbol.
I have an
It sounds like Derrick might have an answer for us.
answer that I really fits my.
So Derrick has an answer he thinks really fits his brand. It looks like some of our viewers at home are thinking we're looking at a leaf or maybe at a grasshopper. Al, let's start with you. Let's reveal your answer. What is it that you think we're looking at?
I think it's a close-up of a classic blade of grass.
Okay, Al thinks it's a blade of grass. Dr. Pitts, sir, what do you have for us? I see a frowny face. What was your answer, Derrick? Could you shout it out for us again?
Of AstroTurf.
AstroTurf. You think we're looking at AstroTurf? I think that's an excellent guess. Okay, so Al thinks we're looking at a blade of grass. Go ahead and push that like button if you think Al's right. And Derrick thinks we're looking at AstroTurf. So go ahead and push that heart button if you think that we're looking at AstroTurf. And before we dive into that final answer, Al, go ahead and tell us why you think we're looking at a blade of grass here.
Yeah, so first of all, the green, obviously, check mark on that one. But then the veins coming, like the lines coming up, blades of grass definitely have those long lines. And I played with a lot of grass as a kid. I loved like sitting in the grass and like ripping it and like looking at it really close and like taking it apart. And I remember feeling the edges being like kind of sticky or pointy. Yeah. And that totally looks like what would have caused that sensation. So that's why that's my guess.
I love that. So digging from your science experiences, that's why we want to explore the world around us. I love that, Al. Derrick, why do you think we're looking at AstroTurf here?
Looking at AstroTurf because it looked green, the side there, and I thought it had some elements of an alien environment. So that's why I went, what, nothing?
Okay, so Al thinks we have a blade of grass. Derrick thinks we're looking at AstroTurf because maybe it has some alien elements. And Al, ding, 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 you would be correct about a blade of grass.
Do I get to do a victory dance?
Yeah, well, easy. No, it's not. We're not done here just now. So Derrick, you heard that Al was correct there. It is indeed a blade of grass. And that experience that they related to us is bang on. What we're actually looking at on the edges here are little tiny deposits of silica. So this is really, really cool. So we know about silica often because it's in minerals, right? Like maybe you've heard about silicates, or maybe you've heard about silicon dioxide, for example, right? Or quartz. Right? Indeed, it turns out that quartz, which contains silica, is the most common mineral at the Earth's surface, but silica can also be taken up via biological processes. That's exactly what plants do. Some plants take up silica in its acid dissolved form when it's still in the soil. It's taken up through the roots, and it's used within the plant structure to either provide some nice rigid structure to it, or a defense mechanism. So in this particular case, it's probably providing some sort of defensive mechanism. Now, there is a really, really cool story behind these features here. These little features, they're called phytoliths. And they've been thought to have spurned on something called coevolution. So during the Cenozoic, this is a time in geologic past, as these grasses started to populate across the American West, we also saw a rise of a special type of herbivorous mammals, herbivorous mammals. So mammals that eat of course, plants, those that chose to eat the grasses, had a special feature. Their teeth grow continuously. So unlike humans, we just have our adult teeth grow in. Some mammals have their teeth growing constantly. And it's perhaps some evolutionary biologists think, in response to eating foods that wear down their teeth, like those that have silica in them. Okay? So
You're blowing my mind, Rachel.
I know.
This is so cool.
So certainly doing the Franklin Outside series has been an opportunity to delve into all of the natural sciences. So a little bit of geology, a little bit of biology, a little bit of evolutionary biology as well. So that's pretty cool. It also turns out that silica can disrupt the digestive tract of either those mammals or even smaller insects. So it might be that defensive measure has to do with how the silica is digested, or rather how difficult it is to digest for insects and herbivorous mammals. It's pretty wild,
cool.
Isn't it?
I love that. And something that, as people who know me know, that I love snails and things like that. One of the main things that I love about mollusks is biomineralization, their ability to, make that shell. We biomineralize and make bones. I didn't know that grass basically did that with silica. Like, that is so cool.
It's pretty cool. It's pretty cool. And it does look differently if we could get even closer under this microscope. It's crystalline structure. So when it's taken up by plants, it actually doesn't follow the crystalline structure, that atomic structure that quartz does. So when we pick up a rock that has quartz in it, it does actually look different than what's called biosilica. So that's the story on my blade of grass there. So love that. When you pick up grasses and they feel sharp on the edges, it's actually because you're touching those little tiny razor blades there. And that's what earned some grasses different names, like the blade of grass, cut grass, sword grass, or even one anecdote I heard was called rip gut. Early settlers had actually noticed that the guts of their horses and dogs would get cut up by some of these grasses. So isn't that wild?
That's so much wrapped up in that.
Okay, we'll stop there. It's fascinating stuff. Okay, so we're going to switch to our third sample here. We'll come over the shoulder now. All right.
Awesome.
All right, folks, this one might be my favorite.
Ooh. Oh my gosh.
All right, let's get a look in.
I like the look of that.
There, this one's pretty wild. We're gonna start our minute just now. Here we go. And I'm gonna adjust as we go to sharpen that image. And tell me, what do you think we're looking at? And what are the features that you're picking out, Derrick, as you're looking at this?
I'm looking at the individual cell-like structures.
Mm-hmm.
I'm looking at how light seems to come through them. I'm looking at how they're clustered, but I'm also a piece of something larger.
Yeah, you hit on a couple really cool themes there. And so you noticed how the light is traveling through certain materials in there. Obviously, you're looking at the overall morphology or the structure, you said, of those individual cells. We'll reveal in just a minute what we think it is. So we've got about 10 seconds on the clock. So Derrick and Al, go ahead and write your final answers down if you have them.
I have one.
You have one. And you may also notice, folks viewing at home, that there's a difference in how my camera is able to focus on the little individual cells that Derrick picked out. You'll notice that in some camera views, I can focus in on that really light yellow part, that really bright part. But in some camera views, if I adjust differently, I can better focus in on the edges there, those dark edges around. We'll talk about why that is in just a minute, but we are out of our guessing time here. So Derrick, let's start with you. What did you think that we were looking at? What was your final guess here?
My final guess.
Flower pollen.
Oh, that's a good one.
All right, excellent guess, Derrick. Derrick
It's
gets
flower pollen.
Flower pollen. We have some folks at home that have also guessed pollen. Someone has guessed corn. I agree.
At first, I thought it was an alien egg sac.
So we've got pollen, corn, maybe an alien egg sac. Very cool. If you think Derrick
And I
is right,
came back
go.
to something more.
If you think Derrick is right, go ahead and hit the heart button. Al, what was your final guess?
Speaking of alien egg sacs, I'm thinking it's some sort of insect egg sac
Love
that
it.
we're looking at here.
All right, Al thinks we're looking at an insect egg sac. If you think Al is right, go ahead and hit that thumbs up like button. And tell us a little bit more about why we made these guesses. Derrick, let's go back to you about that flower pollen.
As I was looking at this, I was noticing how granular it is and how there are individual grains. And I thought those individual grains could be little sort of seed pins, say like a dandelion before it erupts, or in like, the trees, the seed packet of certain kinds of trees that have the seeds packed together like this. That's what made me.
Cool. I think those are all excellent observations. And of course, we also noticed the yellowish color, right? So maybe that's also helping some folks here think about pollen and maybe about corn. So Al, tell me about why you made the guess around an egg sac, perhaps alien, perhaps not.
Yeah. So one of the things that Derrick touched on was that it looks like it's a piece of something larger. So either that it is broke off of something or it was scraped off of something of that nature. So that sort of took me there. But then also the first thing I noticed is like what looks kind of like air bubble-ish to the left side there, which makes me think that there is liquid, a fluid of some sort supporting maybe something in there. And then the other thing is you said about like being able to focus in on different areas, kind of understanding the depth of what this might be, kind of led me there as well.
Yes. Great observation. And let's talk a little bit about that before we reveal our final answer. So Al mentioned, and obviously they have some microscope experience, as we saw their microscope lamp earlier. They noticed that we could focus in on some features that sort of seem to be maybe in the forefront of the image. Maybe they're a little bit higher. And in other areas, it looks like material is a little bit lower. And that's exactly what is causing the difference between our resolution here. Just like when you use your camera phone to take an image, sometimes you can fade out the background or you can fade out the foreground. Can't necessarily do both at the same time, right? And that's because your lens is focusing in on a particular object at a particular distance. We're seeing that happening here, and we see that happen even with the most powerful microscopes in the entire world. So I'm playing around with the resolution a little bit here, and I'm going to zoom in on one of these little individual structures. Hopefully you can see that well. Some of us perhaps thought this was individual pollen grains. Some of us maybe thought this is an egg sac. One of our public viewers said maybe it's corn. Actually, what we're looking at here is a dried orange rind. So this is the outermost edge of an orange, which is pretty wild, right? So I did bring one along to hold up to show you. Oranges actually do have these pores. We'll look up nice closely here, right? And what each of those little pores contain are actually the essential oils of the orange. So these individual sacs, before they were dried out, used to contain some of those oils. Now the outermost edge is called the zest. So actually when we're cooking and we zest off the edge, which is exactly what I did on the other side of my orange here, that's what holds the oils in. But when the fruit is, if you rub against it or if you squeeze it, if you ever opened an orange up really, really quickly and it's sort of squirted at you, that's the oils that are being released from these individual little pouches here. And they really come pushing out of there. They come out about 10 meters per second when you like squeeze an orange, which for those of us that don't know off the top of our head, that conversion is just about 22 miles per hour. So,
that's so funny.
Some pretty intense pressure builds up there. And this again was an evolutionary advantage.
Really cool.
If animals would have interacted with this, that strong scent, that essential oil being released from the orange, then attracts other animals to come and eat the fruit and deposit, of course, its seeds elsewhere, to establish elsewhere, to grow further abroad. So this is a great evolutionary advantage of the orange.
So cool. And it's so funny that like all the things that we noticed were kind of right.
That's
Just not
Yes.
like there are sacs. There was kind of these air bubble sort of remnants. There was, yeah, that's so cool.
Yeah. Yeah, that is really cool. And I love that's,
Eggs inside.
yeah, it's a point I wanted to really touch on with this. Like when we make scientific observations, all of these descriptive points matter. And again, if you had the sample in front of you, you could also ask, what does it smell like? What does it feel like? Right? So we noticed color, we noticed shape, we noticed that depth of view. So all of these considerations, of course, go into identifying imagery when we're looking at it under a microscope. Alrighty, I'm having too much fun here. So
we are going. Wait, we're wrapping up the third image, which means we're almost done.
We're wrapping up the third image. We've only prepared 4 today, but if you like what we're doing, let us know, and we'll try mixing it up again for you next time. So we do have one last sample.
Cool. Okay.
All right, Derrick is coming into this ready. I can tell you're sort of putting the fighting gloves on here.
Well, you only got one more chance, bud.
Come on.
So one last chance, Derrick, for you to tie it up. And I don't have a tiebreaker
Come on,
here.
give me one.
All right, let's get our final sample on. And our final score as it stands right now is Al to 1. And Derrick, you're lagging behind, man. So if you're coming with it, you're gonna come with it now.
Big goose egg, right.
You're just saving it for this last one. You're like gonna really come in strong. I know it.
Alrighty.
Number 4.
Number 4. All right, let's start that minute clock.
Okay.
And tell me what you guys are noticing about this guy. Let me try and, there we go.
Wow.
Noticing some pores. Reminds me, the shape reminded me of a tooth at
Okay,
first.
porous. It does look like a
It does look like a tooth. I will say, don't let me mislead you. The shape of this sample does not reflect,
tooth, right? Uh-huh.
yeah, it
It's
does
got a
not
sandy
reflect
color.
the overall shape of the object.
Ah,
Hmm.
okay. And the magnification is about the same, around 70 or 80.
We've been just about in the 100 plus range. Actually, I think that we can get upwards of about 300 times, but we haven't gotten anywhere near 300 times just yet, Derrick. Let's try and zoom in a little bit more on some of those pores. Al, you called those pores, right?
Yeah, or little pock marks or something. Is there any way we could get like a little bit of light on another angle, like an oblique angle of light?
Oh, I certainly came ready for that. We can try it. So I have a second light source. So let's see if we can try and.
And when I say that, I mean light from the side for our viewers who may not know what I meant there. I want to get some light from the side to see if it'll cast some different shadows on the thing and give us more details.
So it looks like these aren't making a difference. And actually, you'll notice I'm on my video setting. I have my phone camera on. So I'm going to go back to my photo setting, zoom in again, and you'll notice all of a sudden it got really, really grainy there. That's because I don't have that light on anymore. So now I'm going to hold my lights up from different angles. And again, this is a really interesting feature. We're getting kind of a cruddy image, aren't we? And what we're getting at here is really the artistry of microscopy. Anything that you're looking at in that camera right there is just a reflection of light. So anything that we're determining as a color or anything that looks like a shadow or any way that we can even tell that this surface is, as Al said, kind of maybe has pores or sort of pock marks, it's because of the shadows that are being cast ultimately from our light source. Actually, I think the best one that we did have there, Al, was that first setting. So I'm going to go back.
Yeah, I think you're right.
To our video here. We'll try and zoom in again, and I'll leave that.
You have led us astray.
No, not at all. So let's try there.
I think I had the perfect guess, but I've been thrown off now.
So it looks like someone in our audience had a piece of a gingerbread cookie, and you know, I could sort of see that. It kind of looks like it's
still cleaning up leftovers from the holidays.
Oh, nonsense. Those were gone ages ago, Al. Christmas cookies, holiday cookies don't last very long in this house. So I think we'll wrap it there. Our panelists got plenty of extra time this time around. And if you'll remember, Al is up 1-0. Derrick's 0. So let's see if we can change up our points. Derrick, let's start with you again. What do you think we're looking at here? again on the edge of my seat.
Okay, so I've come down to, it's a tiny piece of cheese.
But what kind? What kind of cheese?
I'm sorry, camembert. I forgot to mention.
Camembert. Okay, Derrick thinks we're looking at camembert. If you think Derrick is right, go ahead and push that heart button. Al, are we looking at cheese? What do you think we're looking at?
Cheese definitely crossed my mind, but I think that it is a chip of marble.
A chip of marble, very
a little
specific,
piece of marble
and also
rock.
yeah, also very poetic there. A chip of marble.
Wow.
I really like that. All right. All right.
Yeah, I'm wearing my poetic glasses today.
So Al thinks we're looking at a chip of marble. If you think Al is right, you can push that like button, that thumbs up. And if you think Derrick is with the camembert cheese, camembert or non, Derrick thinks we're looking at cheese, you can hit that heart button.
But I
We have a disclaimer, Derrick.
think you need to change your glasses.
Al, Derrick thinks you need to change your glasses. I actually left mine off today. I was creating too many reflections. So kudos to you guys for having figured out how to do this without having the white eyes.
Yeah, Derrick, I think you need to borrow my glasses because I'm the one that got the point earlier. So
A lot of trash talk. There's a lot of trash talk here. This is a family program, need I remind you folks. So we've got a lot of audience feedback here. Someone says they think we're looking at bone. We heard gingerbread cookie earlier. And someone says a potato peel.
Yes.
Potato peel, I think, is a really good guess too. So before we reveal that, then top line takeaway, Al, why do you think that we were looking at marble?
So the first thing that we saw when you kind of first got it on screen was the shape, which I said reminded me of a shark tooth or something. But it really just looked like a broken piece of rock. But as we zoomed in, it didn't look grainy or anything like sedimentary rock. It looks really like stuck together. And so marble and other rocks that are metamorphic that form under a lot of pressure, that is what it reminds me of. Those like little pock marks is what gives me that image.
Cool. Love it. You know, I'm a geology nerd over here. So Al thinks that we're looking at marble probably because it looks a little bit smooth, but it has some pock marks in there. And Derrick, could be cheese. Tell us your top line takeaway why you think we're looking at cheese.
The top line reason I think we're looking at cheese is because I see what looks like some sort of an outgassing process here. And so I would have gone with something more geologic that you might find on another planetary surface, but knowing that because of the way the surface appears.
Okay. All right. So Derrick went with cheese because he thinks it has maybe something to do with outgassing. Well, I will say that outgassing is related to the shape that we're ultimately looking at here. But no, it is not thanks to cheese. And no, it is not due to metamorphism. So both of you, goose egg, big old goose egg. Or actually, I should say chicken egg because we're looking at a chicken egg shell.
Oh my God.
And I've brought along another, isn't that wild? I brought along another large piece. You can sort of see, you can sort of relate now to what we're looking at, right?
There we go.
It's pretty wild. So when I first pulled this up, I thought it looked like blackheads on skin, which is pretty
it kind of does,
right? But what we all picked up on there, and in different ways. Remember, I thought it was blackheads. Maybe Al thought it looked like rock. Derrick thought it had to do with some sort of gaseous process. All of those individual processes would indeed need some sort of little pore for them, right? So we're all picking up on the fact that this surface has pores in it. And chicken eggs, and it turns out avian eggs or bird eggs in general, for the most part, have little pores like this. In fact, they can have up to 17,000 individual pores on the outside of shells. And what these little pores allow for is gas exchange. That's what I was mentioning. It had to do with that word outgassing that Derrick mentioned. The gas exchange allows both air and carbon dioxide to enter and exit through the eggshell. So just like So when humans before humans are born, we get oxygen through the umbilical cord, of course, connected to our mothers. But chickens, of course, and other species that hatch from eggs aren't connected to their mothers in that way. So how do they breathe before they have lungs, before they hatch out of the eggshell? Well, it's due to this gas exchange, and it's simple diffusion, which is amazing. All that means is that air is passing freely from the outside of the shell to the inside of the shell. And on the inside of the shell, there's another extra membrane that basically makes up a blood vessel network that the oxygen can diffuse into. It's delivered to the embryo, and it cycles, those blood vessels cycle that material towards the edge of the shell and then back in towards the embryo, where they can release CO2 when they need to, and they can pick up oxygen, come back into the embryo, come back again, and that process starts all over again. So that's what these little tiny micropores are on the outside of our chicken eggshell.
That is so cool. And again, it goes to show with this sample that, like, I thought it was flat. Like, my perspective was that it was totally flat. And now that you show it, yeah.
Yeah, agreed. It is really, really tough to see that we're looking at a slightly curved surface. So Derrick, you cut off there. Was there a final observation you want to share?
Yeah, at home, you might think the surface is very smooth, but under magnification, that belies
Yeah, Derrick, I agree.
something really important about it.
Yes. Yeah, magnification does reveal all of these really important features that are invisible to the naked eye, and they're maybe even invisible to our sense of touch. We might be able to feel those little tiny pores. I can't. I have damaged fingertips a little bit, but maybe someone that has very, very sensitive fingertips could. But I agree with you, Derrick. We need that power of magnification to really tell just what it is that we're looking at. So I'm going to stop sharing this guy here, and we can wrap up our video feed with just the three of us. Well, thank you, Al. Thank you, Derrick, for joining me today on our Franklin Outside live stream. Of course, we were inside today, but we're looking at our outdoor nature samples. And Derrick, I don't know, that looks like a celebratory dance, but our champion today was Al. So Al, give it up.
I was gonna say I should be doing the shake, but I'll let you do it too, because I wouldn't have been able to do it without you right there by my side, buddy.
At the spaciest answers of all. Thanks, Al.
All worthy answers. Thank you very much for making very real and really substantive observations as well to share with our audience at home. So for those of you watching at home, of course, if you liked today, let us know, and we'll continue trying to do this. Al, Derrick, I hope you guys had some fun today, and maybe we'll see you back next time on Franklin Outside.
Thank you so much for having me.
Thanks for joining, everyone. We'll see you next time.
Thanks for having me.
Bye, Derrick.